So I will talk about a project that we started a couple of years ago. And it's a community resource for bioimage analysis training. And I assume since you joined, maybe it's interesting for you to also teach bioimage analysis. So if that's the case, then maybe this is interesting for you and maybe you would like to join this effort. So we are very open for everyone to join. So let me start with a motivation. So why did we actually do this? So I think the challenges still are, and definitely, I mean, as mentioned, I think we started like 5 years ago or something like that. So I think landscape might have changed a little bit, but I think not too much. So there is still, I think, quite a high demand for bioimage analysis training. So I just asked actually a colleague, they run every year an international course, and they typically have 4 times more applicants than they can take. Then at the same time, why don't we just run courses all the time? Well, it's actually quite some effort to prepare a course. And that's where we kind of started. Why is it so much effort? I think the reason is that still typically mostly each of the trainers that you invite to give a course, they have to prepare the teaching material. I mean, they might have some, but then they need to update it. And then, so it's still always quite a bit of work, I think. And the other reason why it's a challenge is that you always might want to teach something slightly different, right? There's really so many platforms in which you can do bioimage analysis. And then for each of them, you have then basically to come with different teaching material. So that's another challenge, I think. So what we try to do is have something on the web that is really reusable. Okay, so basically we use the same training material that we used for one course for the next one. And that's not so easy actually to do this. So how we try to make it reusable is that it's actually a collection of modules. Okay, and each module is relatively small. So you can teach a module in something between 20 to 40 minutes. And then you can package them into courses. So then you can decide, let's just teach these 3 modules or maybe 6 modules. So you can kind of very flexibly design your course. That's the idea. Then we already used this teaching material for both online teaching. So we actually used it during COVID quite a bit and also in person. So it works in both contexts. And then what we try to address is this issue of having all these different image analysis platforms by having a separation for each teaching module of the concept that you want to teach. For example, image thresholding. Okay, this is something that is just, you can actually describe on a whiteboard or in a textbook. You don't need any software for this. So this is one block, and then as you will see, we have then specific implementations how to actually do it. And like that, like in Python, for example, or in ImageJ. And like that, it's a bit less effort now to add, for example, if we now want to add teaching material for teaching thresholding in Julia or something, it's very little work because we just have to plug in the code, but the whole framework of explaining everything else is already there. So you will see that. So it's actually really pretty efficient in terms of just adding yet another software in which you would like to do this. Okay, and then this is hosted on GitHub. And so there is all the GitHub convenience of issues, pull requests, and so on. So it's quite nice to continuously improve this material and add new things. So this is like the overall idea. So, and I will maybe now immediately jump to the website. So we have now 37 of these modules. 3 of them are about workflows and 7 of them are about scripting. So I will just go there. And if you would like to go there as well, if you just Google Neubuyers and here training resources, I think that typically the first hit should get you to the GitHub. And then from there, you would have to go to a link that's in the README. It's quite obvious, I think. So basically, here's this website, and we have all these modules, okay? And they all have this title and a little image with them that describes what it is about. So I will just slowly scroll through. As mentioned, these things we try to keep kind of small concepts, okay? Like what you would like to do. For example, you want to filter objects in an image, or you want to perform a distance transform, or you want to do a watershed. And there we try to explain these things on their own. But obviously, it's also important to know where they belong in the whole image analysis workflow. And this is also why we have some workflow modules where then basically some of these concepts are combined to actually get some task done. And in fact, these workflows, I think this is only 3, as you see. So this is definitely something where I think it would be really good to add a couple more. So if you are, after this talk, motivated to contribute, that would be a great point. Then we have decided to do, I mean, this should not be really a programming course, but maybe it is now. So some specific modules for scripting image analysis, okay? Because that's obviously very important if you want to do something reproducible. So here we have now some modules about that. And in fact, these are actually only implemented for ImageJ macro at the moment. But as mentioned, I think it would be not much work to add Python to this. And I think that's something we will do in the near future. Okay, and then there's here a bunch of draft modules. This is basically things that we started doing at some point, but that are not completely finished. But we thought still, let's just put them out there that we don't forget that we have them already. Okay, so this is how this looks like. Then each module has different sections. And I think I will simply go to one of these modules and just show you live. So, for example, the thresholding module. So now let's say you want to teach this, okay, as a teacher. So first of all, what you can see here is in the top prerequisites. So there are links to other modules that people should know before taking that one, okay? So this is giving you an idea of knowledge that you should have before, and that can also help you designing a course, right? So maybe then if you design a course and you want to show thresholding, you somehow should check that people know these things. Then we have always these learning objectives. So after completing these lessons, the learners should be able to do, for example, here, describe the relationship between an intensity image and a derived binary image, apply a threshold to segment an image to foreground and background regions. Okay, so this is what you would like to achieve. And maybe I have it in the acknowledgements, but maybe I mention it here. So this whole website layout and also all these boxes is actually to a big degree copy and pasted from a consortium that are called the Carpentries. So you can Google that too. And these are really people that design excellent teaching material. And they have also trained the trainer courses. So I highly recommend having a look there. And actually, the whole reason why what I show you today exists is that I joined such a trainer course from the Carpentries. And I was really impressed by the quality of their material. And then I thought, let's try to do something similar for image analysis. Okay, so this is really, and actually we got help from one person that now works full-time for the Carpentries. So this is really not our thing. We just copy and pasted their website basically. Okay, so then we always have this motivation thing, right? So it's important, I think, to tell people why should you learn this? Okay, and then there is this concept map. And that's interesting. That's also something I learned from the carpentry. So here, for example, the concept is you have an image, you apply a threshold, and then you get the binary image. And the point of a binary image is that it has just two values, either it's a background pixel or a foreground pixel. So this can be useful maybe already as is for explaining, right? So you can show the students, okay, this is actually the point. But actually another reason to do this, and this is what I learned in this Train the Trainer course, like if you teach, you should, for each block that you teach, that is maybe 15 to 20 minutes, it's very useful for yourself as a teacher to draw such a concept map. And the idea is if there is more than 5 to 7 words that the students have not known before, then it's too difficult, probably, okay? And this is based on some psychological research, actually, like how much can you actually keep in your brain in terms of new information? So this is something I learned during this course. So this is also basically for the teacher when designing new material to check that this is not too much now, right? That this is a digestible amount of information. So this concept map also then, as mentioned, is for you as a teacher also good to limit yourself and not go astray. Okay, and then so each module has a figure, and so the purpose of this figure is really to try to summarize more or less everything that you want to teach. Yeah, and that's of course very challenging, and we don't always manage. And I think all these figures that we have can be improved, and again, ideas and contributions are welcome. But for example, now, if I would teach this teaching material online, I would spend quite some time on this figure to actually explain what's going on there and the concept of a threshold. If I'm teaching in person, another thing you could do is actually explain whatever is in the figure on the whiteboard. Okay, so then I would not necessarily actually use the figure at all for the teaching, but just for me as a reminder what I should roughly get across on the whiteboard. Okay, so in that sense, this teaching material, as I mentioned, it's quite flexible. It should just help teachers to give a course and don't have to prepare too much. Okay, then once all of this is explained, then actually we have this activities block for each module. And there are several activities, and actually for me, now, I don't know how old or young are you, but this whole thing reminds me now of my math textbook at school, right? There was always this upper left corner with maybe some figure and some general explanation, right? And then there were 1.5 pages of exercises, and the teacher would do one of them with you, and 3 of them give you as a homework. Okay, so this is how all my math school books looked like when I went to school. And actually this is now very similar. So I don't know if that's a good or a bad thing. But so basically one can now choose these activities, and then as a teacher you can decide to do maybe 1 or 2 of them, either demonstrate them or do them together with the students. So do it so slow that they can follow along. And then you could say, and then maybe now tell them, look, this one maybe try on your own, and it could be even during the course, then you give them some time, or it could be some homework until the next day or something. Yeah, so this is also really up to you as a teacher. And now, as I said, the interesting thing I think here is there is this task described, but just in words, without mentioning any specific software. Okay, so this was the idea. And then you can choose now different software platforms in which you really want to do it. Okay, and so for example, here is the ImageJ GUI implementation of this task. Okay, so you open this image, and then there's very specific stuff what you would have to do in ImageJ to do the activity. And as mentioned, typically I would, if I have enough time, I would do this slowly and tell people, okay, just click along with me, right? But you could also do it faster and just demonstrate it if you have less time. And then what we did recently, actually, just a few weeks ago, for some of the modules, we added now SK image Napari implementations, because we think that's an important platform. So here is basically Python code doing the same task. And here again, how you teach this is, I mean, it could be, for example, telling the people, copy and paste this part into some terminal and execute it. I mean, it's a scripting language, so you could do it step by step. What we decided is to open all of this in the Jupyter notebook, and actually these are special comments that are kind of cool. So if you have the right extension installed, it's called JupiText, and if you open this thing in a Jupyter notebook, it will break it into cells here, so which is quite convenient. So you have then the thing both as a normal script, but it would also render nicely as a notebook. Okay, and then here's now a lot of code, of course, but then people should also do stuff in Napari, which is the image viewer that we're using, and in that case, the instructions for what to do in Napari are here as comments in the script. And then you don't forget that this is what one should do. Okay, so this is basically this, right? And as I mentioned, then these modules have more or less of these activities, and you can decide how much time you want to spend or how much you want to give as a homework. Then in addition, you can let the students do some assessment that is, again, independent of any programming language. And these are basically just some quizzes that prove so it's called formative assessment in the carpentries language, that they're basically like the stuff sticks better in the brain. Yeah, okay. So for example, pixels in a binary image can have maximally different values, and then you should know that should be 2, okay, either true or false. So basically then telling people, please do this, and is also nice. In general, what I find nice is if you tell participants to do something, is to tell them to work in little groups. So I always, when I teach, tell them, please work in teams of 2. And then only one of you is allowed to touch the computer, for example, when it's something like this, and the other person should make interesting comments. So basically that it gets very interactive. So this group work, I find just as a general information, I find nice, then it gets also more lively. All right, then for some modules, we have also something called explanations. And that would be text that's needed, I think, if you don't have a teacher around, okay? So that would be then, like, if we would add that more systematically, it would allow also for people to use this material for self-learning. But initially, our idea was not that this is the use case. It was really meant to be a resource just for trainers. But the framework is there, so one could decide actually to add much more in this explanation text, so that it would be easier also for people to use this for self-study. So again, if you think that's interesting, that would be very easy to add. Then the last thing is, so what comes next? So there's basically modules that we think make sense to teach next. So again, if you design a course, maybe you could look here and check whether that makes sense for you. And then we have also this learn more category, where we basically can add links to any other resources that are doing a good job at explaining whatever is the content of this module. Okay, so that's basically it. So this is how all these modules look like. So I can go back to my presentation. So here it's the same again, just with screenshots. So I went through this now live. Okay, so about the development of this material. So it is currently at 19 people have contributed, and this is just by looking on the Git contribution. And But obviously, I mean, if you go there, you see some of them maybe only added one typo. So, but some others added substantially more. So I would say really like, I don't know, something between 5 to 10 people really added quite a lot of stuff. And these were also the people that then actually were teaching in courses. What I find nice is that this is really now spanning several institutions. So not only EMBL, where I am from, but also one person from Max Planck Institute in Germany, and then another one from the Friedrich Miescher Institute in Switzerland, and also from other institutions. So, and actually these 3 institutions that I mentioned here, we were teaching a course together with this training material just a few weeks ago, a Zoom course, and that was very nice because then we can really just share the labor, and it's also much more fun, right, if you develop this stuff together. Yeah, and then the contributors are mostly staff scientists, right, working in microscopy facilities as bioimage analysts, so sort of professional people. And really, so what we actually try to do is now since a while, so like every last Friday of the month, okay, we try to spend half a day working on that stuff. And then the people, like when we have a Zoom call together, and we really discuss quite a lot how to teach this and what the content should be and so on. And we are also changing stuff all the time. And it's also really, I mean, the tricky part is, and there's really not only one way of doing this. So it's very difficult sometimes to decide on something. And then when you changed it, you want to change it back and so on. Then here is a link. So the presentation, I think, will be later shared, but you can also go to the Git website with just some specific tips that we recently added for people that want to teach this material, what we think works well or doesn't work well. But I think I mentioned some of these things. Okay, so what did we actually do this? So, okay, I exaggerated with 5 years. Okay, so apparently we only using this since 2021. But we already ran 9 courses with this material. And when I say we, it's really, so it's me, but also mainly also so his name is Antonio from Max Planck Institute in Germany. The courses typically that we ran with this took between 1 and 3 days, and we had in total more than 300 participants there. And then, so the material now, it's mainly for beginners. We have some modules that I think a bit more advanced, like distance transforms, watershed, and so that's maybe not something you want to do in a beginner course. And we have also the scripting modules. And then in terms of the platforms, we mainly have implementations for doing it in the ImageJ graphical user interface, or using the ImageJ macro language, or now, since very recently, also using the skimage Python library and Napari as a viewer. So maybe I want to click on this link. So this is the most recent course that we taught. So this is also here on the website. So it was this beginner's course for Python-based image analysis using skimage and Napari. So this was 3 days, for example. And always what we decided to do in this case was to have in the morning sessions where basically we only demonstrated stuff using this material in Zoom. And then in the different institutions that were participating, we had then rooms booked where people could then try to repeat what we did in the morning on their own in hands-on practicals, and there were teachers around to help them. This was something we tried to accommodate the different institutions. So there were actually a bunch of teachers involved, as you can see here. And here you can, for example, see, right, how we decided to do it. Maybe it's interesting. So this is what we decided to teach on day 1. And then on day 2, this was the content. So I just give you time to read it. And then we had a 3rd day where but this was the covered content. And then I think we covered already quite a bit of the basic stuff that you would like to know if you do image analysis. So that's an example, right? You could design other courses that are shorter or longer. Okay, so in terms of future directions, so one can always improve the material that we have. So for example, I think what's not so good right now is that some of the example images that we are using to teach a specific concept, they were really chosen to be the most simple image that you can ever imagine to to get the point across. And I think that has a value that's useful, right? You don't get people distracted, but then there's no biological context somehow. And then I think that's not ideal. So it would be nice to improve the example images somehow, that they're still simple, but that you can also tell an interesting biological story. So I think that's something where we could put some work in. Then, as mentioned, we could just add more text. We call them explanations for self-studying. Then also just always add more modules and specifically workflows. And I think the workflows are good. And that's something also where I think we can still improve when we teach is, I think it's actually very good if you maybe start with a workflow, right? And say, okay, this is all the things that I would like to do to answer my biological question from the beginning to the end. And then you can easily, it's much better to motivate why then you have to learn all these individual steps. And also when you are teaching an individual step, you can always go refer back to the workflow and say where you are right now. So this is something which I think we didn't do enough, which would make the teaching much better. But for this, obviously, we would need maybe 1 or 2 more workflows that we are sure we really cover all the modules. Then a bit of a bigger project is, I think, but it's very straightforward because everything is so structured, is to actually record YouTube videos as teaching all of these modules, and then people could watch them also without someone giving a course right now. So that could be done in a very straightforward, systematic way. And I think that's something we want to do. And then another thing we would like to do is actually another big challenge if you do a course, I think, is finding good example data, at least for me. And we have some example data right now, but actually all of this is in the GitHub repository, which is something you should not do normally. Okay, there should be only text, but we specifically chose very small images. And I mean, the nice thing is everything is in one place, but I think it would be interesting also to explore having really one central public place, and we think Bioimage Archive is the place where we have a bucket for data that's useful for training courses in bioimage analysis. So this is something I think also we are looking into. Okay, so that brings me to the end. So here is a list on the left of all the contributors, excluding myself, and they are sorted according to GitHub commits. So Antonio had the most commits, and then Toby, he was actually the person that helped setting everything up, and he works now full-time at the Carpentries, which I mentioned already. So check that out. That's really interesting. And I would like to personally acknowledge the Chan Zuckerberg Initiative for funding myself at the moment. Okay, and that's it. So I'm excited to answer any questions.
Okay, thank you. Thank you, Christian. Thanks a lot for today's webinar. I think it was very insightful, and it's a great open resource which anyone can use. And as a trainer, I can say probably I mean, everyone has their own style of teaching, but I think the bigger effort is like creating the content for your teaching. So you don't have to create things right from scratch if you are planning to teach bioimage analysis to anyone. So I think it comes, and this resource comes in very handy. And if you have something really nice that you have developed, and always you can get in touch with this resource to contribute to this. All right, and thanks everyone for joining in. And so we'll take the questions now. So this is time. If you have question, please type in your questions here. Okay, so we have one question here which asks, what are different characteristics between common images, medical images, and biological images?
Yes, it's a good question. So what's like, for example, if you want to teach, like I think some modules that would be the same are what is a digital image. So this is typically our first module because all these images are digital images, right? So you can teach that they have like pixels and coordinates and stuff like that. Then they also, like the data type module would be useful because they have different data types, but some of them are maybe RGB, others are 16-bit and so on. Then I think teaching modules about how to interact with multi-bidimensional images can be interesting. For example, they can have 3 dimensions or can be actually have multiple colors, like if you think about histology. I think also the segmentation concept, right, that you want to find objects in images are similar. So actually, I think there are many of the teaching modules that you could probably reuse. I think we discussed this recently. I think one thing that's, for example, we don't have right now, and that's a bit specific to medical images, is a teaching concept. I think it can be called color spaces. So we have mainly started from fluorescence microscopy images, and there you just acquire 1 or 2 channels or something, right? And you treat them as grayscale. But with, I think, for example, medical, if you think about histology, then you have these color cameras. And that's a whole different topic, like how the color, right, comes in from these stains and so on. So this is, for example, we are missing right now because we didn't think about it, right? That would be one example where I think there's right now a teaching module missing that specifically deals with this RGB type images. Yeah, so that's maybe one answer. I hope that's good.
Thanks. So you mentioned some carpentries. Do we have some link for these such carpentries within this GitHub document, or?
It's in the presentation.
Okay.
I think if the presentation is and it might be also in the GitHub. I don't want to browse around now, but it's in the presentation, and I think this is shared later, right? Yes.
Yes. So I'd like to let everyone know that I'll share the presentation slides as well along with the recording of this video. So you will get the links.
You just Google the carpentries computer teaching or something. I think it should just be the first hit.
Great. Next question is, thanks for the talk. Can you do everything that you can do in ImageJ also in Python? And if that's so, then is it better to go straight to Python, or you need to know how to use ImageJ?
These are eternal questions. And we just had them recently. Like, we were thinking, what should we actually teach people? So it's a very complicated question. So I think if you are comfortable with scripting, and then I think I would recommend going to Python. And the advantage of doing the scripting with Python, if it's reproducible, right? Like if you have a script, you can publish it and you can share it. And also if you use Python, I mean, it's a huge ecosystem, so you can have stuff for tables, you have all the deep learning things. So basically, I think you can probably do most of your scientific career just in Python, whereas ImageJ is very limited to images, and also the scripting languages that it comes with are much less standard than Python. I mean, there's this ImageJ macro language, but that's very specific. And then Groovy is another scripting language that's actually quite a popular scripting language. So that's a good one. You can also script ImageJ in Groovy, but then again, I think most of your colleagues probably will do Python. So I don't know. I would probably recommend the Python ecosystem and use Fiji mainly for just quickly inspecting some images, because I think that's still not so easy in Python. But it's a very hard question. I mean, I don't know. Also, maybe just do what your colleagues do, because then you have friends that you can ask for help.
Thanks. How complicated is it to add a new module? could you maybe quickly show a PR where one module is added? I'm just wondering how high the effort might be for adding modules in this resource.
So I don't think we have a PR for, so adding a new module, I think is actually a bit difficult. So there I would say it's better to raise an issue and describe what you want to do, and then one of us would just do the initial stuff that's needed to add a new module, okay, and then we would just point you to where you can add the content. So I would not recommend just adding a module on your own right now. So I would rather raise an issue and say what it is that you think would make sense, and then I think one of the people that did that already a couple of times would get in touch with you and help you. So I think that's better, and we would be very happy to do this. But a bit more technical if you clone that whole thing, okay, and then on the command line here's actually a script that's just called add new module, and if you execute that and give it as a parameter the name of the new module, it will already do all the, like, create all the files and so on. So in principle it's easy, but still I think it's even easier if you ask us for help, and we are happy to help.
Right, thanks. Next question is, do you do assessments during in-person courses? If so, what do you use? For example, Slido, etc., or something else?
So we actually just actually just this material. We just tell the people, look, here's the assessment, go through these questions and maybe take, and I think usually we actually never said anything specific. I think most of them take a piece of paper and just write down the answers, and then they check if the solution is right. So there we have no additional technology.
All right, so one question that came in chat actually. So can you use images from your GitHub pages for publication, or do they need any permission to use?
You mean from whether you can
images for publication? Yeah, images from GitHub.
This from our GitHub. I mean, I don't know from GitHub in general, but I guess the person you mean from, like, in general, I have no idea. For our images, they are here in this folder called image data, and I think you can do with them whatever you want according to this license file here, which is very permissive. So yeah, I think you can use these images.
Okay, great. There's one question which I'm not sure if it's very clear to me. What would you recommend any user as a starting point, where somebody who is used to use ImageJ for image analysis, but now they want to change to Python-based analysis, and they would like to change the codes, etc. So what would be the starting point for such user?
You can try ChatGTP. So if you tell ChatGTP to rewrite an ImageJ macro and you give it the code into Python, you get some sort of reasonable script. Like with ChatGTP, as always, it might look super reasonable and then not work, but that could be actually, I think, a really good practical starting point, and then you can try to get that script running, and probably maybe with some help from someone close by. But if it's really specifically converting ImageJ macro to Python, I would maybe really try this. Yeah, just ask ChatGTP to transpile it for you. Otherwise, I think it's very difficult to figure out what is what. I mean, then, in fact, our teaching material might be not so bad, because if you, for example, go here into the thresholding step, I think you could actually look, right? How does this look like in an ImageJ macro? Okay, and how would I do exactly the same thing in Python. So maybe actually this teaching material here could be also a good starting point, because for many things you have both things side by side, and then you could learn how it translates.
Great, thanks. Yeah, so you could come to this module before you ask ChatGPT. I guess that could be a starting point.
Or compare what ChatGPT thinks and what we were thinking.
And please provide the feedback. So what you get.
Okay, let me also say in general, of course, like if there's tons of like if you like for Python-based image analysis, I mean, I don't know now by heart, but I think you will find a lot of teaching material. I don't have a specific tip now. Yeah.
I think we have one interesting question in chat, and I think it's more about data integration, I think.
Mm-hmm.
But the person is asking, how can we concatenate bioimage analysis to genomic analysis? Has somebody tried to overlay these two different analyses for their research-based questions?
I mean, there's two things that I'm aware of. I mean, first of all, there is this whole spatial omics technology, right, where people do find RNAs in image data. So you have this barcoding approach. So spatial omics would be one way. I think the other approach I'm aware of is that people find interesting cells in images or on the microscope, actually. Okay, and then there's different ways of getting those specific cells that look interesting in the image out and sequence them. So that's the other way, I think. Basically, you first take an image and then you have some way to mechanically take that cell and do genomic analysis on it. So this is, I think, the two avenues I'm aware of, basically.
Yeah, that's very good. I think there are very powerful flow cytometers as well these days, where while they are sorting, they're also taking images and they sort cells according to those images. So I think those can also be used.
I agree. Exactly. I forgot this. Yes, this is another one. Yes.